Now it is my pleasure to turn over the program to Dr. Chattervidula.
Thank you, Dr. McHugh. It's my pleasure to introduce Dr. Mark Lovern today. Thank you, Dr. Lovern, for accepting our invitation. Dr. Mark Lovern joined CERTARA, then Quantitative Solutions, in 2012, and has 20+ years of experience in applying modeling and simulation tools and techniques towards optimally informing drug development decision making. Throughout his career, Dr. Lovern has led and contributed to numerous consultancy projects involving population pharmacokinetics, pharmacodynamics, and model-based meta-analysis. Dr. Lovern currently leads CERTARA's Quantitative Sciences services for the United States. His previous work history has been split between biopharmaceutical companies, GSK, GlaxoSmithKline, and UCB, and companies that support the biopharmaceutical industry, such as Quintiles and Farsight. In addition to modeling pharmacokinetic and pharmacodynamic data across a wide variety of compounds and therapeutic areas, Dr. Lovern has taught over 50 technical training workshops on modeling tools and methodology. His most recent therapeutic area experience has been with therapies for infectious disease, metabolic, and autoimmune disorders. Dr. Lovern was awarded a PhD in biomathematics from North Carolina State University in 1997. His favorite leisure activities include hosting dinner parties, traveling, reading, and enjoying films. I welcome Dr. Lovern for the talk and to UNTHSC. Over to you, Dr. Lovern.
Thank you, Ayappa, for that introduction and also for the opportunity to speak today. So I am going to speak about applied meta-analysis in drug development, and we're just going to go ahead and launch right in. So first of all, let's just talk about what I'm going to talk about. So the main ideas today are we're going to talk a little bit about model-based meta-analysis and how it relates to other forms of meta-analysis. And in essence, it's an extension of a technique called network meta-analysis. Oh, crud, sorry, that was an accident. We are also going to talk about why it's useful to make use of publicly available information. And one thing, it basically is much cheaper and to glean information from publicly available information, and is also considerably faster than generating proprietary data in many instances. We're then finally going to close with some sample applications of model-based meta-analysis, including evaluating therapeutic performance of a drug relative to existing therapies, quantifying the impact of patient or trial design characteristics on the magnitude of drug effect, and also characterizing relationships between short-term biomarkers and longer-term clinical outcomes. We've got a pretty good sized group today, but I would love for people to be interactive if possible. If you have questions along the way, please feel free to raise your hand. Virtually speaking, I think there's a way to do that here in the Zoom platform. Otherwise, we can. I'll do my best to address any questions during the dedicated Q&A at the end of the session. So, meta-analysis is nothing new, and it is widely used from a variety of perspectives and applications. So these are just some sample papers that illustrate various applications of meta-analysis. At its heart, meta-analysis literally just means combining results across multiple randomized controlled trials, or even observational studies for that matter. But meta literally means instead of analyzing one study by itself, you take multiple studies and you combine that information in some way. And there are several ways of going about that. And as you can see on this slide, there are quite a few different ways that that technique is applied. So why use meta-analysis? Well, one reason is that data is really, especially proprietary data, is extremely expensive. So on the right is essentially what I have dubbed the cash spiral. This is the cycle that that companies use to generate proprietary data. So for instance, you might start by designing an initial trial. So say, for instance, maybe it's a single ascending dose or multiple ascending dose trial in humans. And then you then go on to conducting that trial, and then you analyze the results from that. And that potentially guides the design of the next trial. In some cases, you might have more than one trial you're designing in parallel, but each new trial brings new information, and essentially the results from that feed into the wider body of knowledge of that particular asset. The issue with that is it's really expensive, and as everyone knows, the expenses continue. The costs of drug development continue to rise. They're not going down. So if we can somehow inform the drug development decision-making with non-proprietary data that's available in the public domain, this can greatly abbreviate the churn associated with the generation of proprietary data. So let's give an example of this. So this plot that you see here, if you were to generate all of the data that you see in this graphic, it would potentially cost $7 billion. That's why I basically say here, this is a $7 billion plot. Now, what are we looking at? Essentially, we're looking at the correlation between a short-term biomarker of glycemic control, namely fasting plasma glucose, which is something that generally responds very quickly to anti-diabetic therapies. You can even see an effect after one dose of a drug. And on the y-axis, you see change from baseline in HbA1c, which is essentially the gold standard measure for glycemic control. It measures the percent of hemoglobin molecules with which there's an adduct with glucose. So the more, the higher the HbA1c, the worse the glycemic control. And one of the reasons that endpoint is of such interest is it's relatively stable over a period of weeks. So it's really a measure of sort of ambient glucose, because once the adducts are formed, they're essentially permanent. And the only way that they're sort of taken out of the system is when the hemoglobin molecule itself is sort of turned over, basically retired, if you will. And so it's a very good measure of glycemic control over time. Now, what about the body of this plot? Well, every dot you see here is a data point from a trial where at the same time within the trial, they measured both HbA1c and fasting plasma glucose. And so these are essentially sample means within a treatment arm. And the colors represent different therapeutic classes. So all told, what you're looking at here is data from about 470 trials of new anti-diabetic therapies, accounting for about 170,000 type 2 diabetes patients. All told, there are 35 drugs with 9 therapeutic classes. So if you estimate, for instance, that a single GLP-1 agonist takes about $200 million to develop, then you put all those numbers together, and if you were trying to generate this plot, under your own auspices, you would be looking at a price tag of about $7 billion. So finally, what is this plot really telling us? Well, it's telling us that there is a pretty strong correlation between a short-term biomarker, meaning fasting plasma glucose, and a longer-term response biomarker, which is HbA1c. And more importantly, that relationship seems to be fairly conserved across therapeutic classes. So if, for example, you were getting ready to develop a new antibiotic therapy and you did a initial study and measured fasting plasma glucose, this plot is telling you that that change from baseline in that biomarker, which should be fairly predictive of what you would see in HbA1c at later times. So it could be very powerful for predicting how well your new compound is going to behave relative to existing therapies. So, as I mentioned, generally in meta-analysis, we work with summary level data because we're gleaning information from published sources, and generally speaking, in publicly available information, they don't provide individual level data. By that, I mean, you don't get data for every single subject in the trial. And there's many reasons for this, including some privacy concerns. So in the schematic, each of these little golden trays represents a clinical study, and the little sheafs of red paper on these platforms or these studies represents individual subjects. So each trial has a treatment, and that which is this TI, and then the XI are sort of the subjects or patient characteristics associated with each of these trials. And you can see that different trials have different sample sizes. Some have more subjects than others. One consequence of this, when working with aggregated data, is that you don't want to weight each of these studies equally. Generally speaking, you're going to give more informational weight to larger studies with more subjects because they're a larger sample size. So across these individual studies, we also will have essentially estimates of an overall central tendency for the population, as well as some estimate of trial to trial, variability and what we call heterogeneity, which we'll talk about a little bit more later. So let's make sure that we understand, what sort of data we'd be dealing with. So if we had individual level data from all of these trials, for instance, you might have, a plot of a covariate and a parameter such as clearance, and the covariate might be something like body weight. Well, you would have a observation for every single individual in these trials, but what we're usually dealing with or have available is summary level data, as I said. And so here you have a situation where for each of these trials, you have but one observation as opposed to multiple observations. And notice also with regard to the covariate, you don't have as much of a range. And that's because you're really talking about the mean of the study subjects, which is not going to be as variable as the individual values themselves. So, as I mentioned, meta-analysis has been used for quite a while. And historically, it has been used very frequently for drug evaluation. So this is often used by regulatory agencies or payers when evaluating how a drug relates to standard of care. And this one, or it can be reevaluating, for instance, how a currently existing therapy is performing and what are its relative benefits and risks. As an example, about 10 years ago or so, there was a class of compounds called COX-2 inhibitors that were used to treat chronic pain. They were non-addictive, they were non-opioid treatments. But then when the sample size got large enough and people were doing meta-analyses, it came to light that there were cardiovascular risks with these compounds, and that may not have been found in the initial pivotal trials because the event was rare enough that it wasn't really shown to be statistically significant until folks started looking at a much larger data set using meta-analysis techniques. More recently, and this is where some of the key applications of model-based meta-analysis is in actually drug development decision-making. So before the therapy has been released. So common questions that we try to answer while the drug is still being developed is, what is the therapeutic window in comparison to placebo? And how does the therapeutic action of the drug compare with existing therapies? Is there a shared dose response curve within a particular drug class? Which dose, regimen, and route optimizes efficacy and safety profiles? And how do effects develop over time, and how do endpoints compare with one another? So let's have a look back at the history of meta-analysis. The term was originally coined in 1976 by a social scientist named Gene Glass. And he was speaking to the American Educational Research Association in San Francisco. And traditionally, social scientists have made major contributions to meta-analysis methods, oftentimes because social science is, it's maybe not as well funded as a lot of other disciplines. So oftentimes the studies are somewhat small and maybe not very well funded. And then to make inference across those studies, then often it involves meta-analysis. However, one of the potentials for the earliest example of statistically combining medical data from multiple studies is by Carl Pearson back in 1904, in which he looked at inoculation statistics for typhoid fever in soldiers of the British Empire. So since about the 1990s, meta-analysis has become a cornerstone of evidence-based medicine. And as I mentioned before, was initially used to evaluate the effects of developed drugs. Now, as I've mentioned previously, its popularity in informing drug development decisions has continued to increase. So one thing to bear in mind is that the analysis itself, the meta-analysis, in order to be robust, is really the culmination of a process generally known as systematic review. And so it's, actually the culmination is in the publishing of the results. The meta-analysis is the penultimate step in that process. So which generally starts with a research question. For instance, maybe I want to know how are anti-epileptic therapies performing relative to one another. So based on that question, I then design a study protocol, which essentially says, well, what am I going to consider in this analysis? What am I not going to consider? So using, again, the example of anti-epileptic therapies, I might say to say in my design, I'm not going to consider therapies that are being used off target. I'm only going to consider therapies that were specifically designed to treat epilepsy. So, so then the next step is a search, which then you perform a literature search. Oh, and part of the design protocol is defining what the search terms are going to be for that search. So, and what, again, and so then you essentially select the retrieved references, and then you do a sort of assessment of bias for the sources that you found. You then extract that data, and then you proceed to the actual meta-analysis, and then generally you publish the associated results. So, as I mentioned previously, we are going to talk particularly today about applications of model-based meta-analysis. But let's take a moment to understand how model-based meta-analysis relates to other forms of meta-analysis. So one of the earliest forms of meta-analysis is what's called a pairwise meta-analysis. So essentially, what that means is that you would combine studies which looked at the same or a shared set of interventions. So for instance, maybe you had 3 different studies that that all looked at drug A versus drug B, and they may have different sample sizes, and so, but somewhere in there you compared these 2 treatments. And so essentially in pairwise, you basically take all of those together and treat it as one big trial, but while accounting for some of the distinctiveness of each of the individual trials. So for instance, it might be in slightly different populations, or they might be in different regions. So network meta-analysis is an extension of pairwise. And what network meta-analysis essentially does is it says, okay, well, maybe I have studies. I'd like to understand how drugs B, C, and D compare to one another, but I don't have any trials where I've directly compared those 2 treatments. I do, however, have studies where each of those drugs were compared to placebo. So by extension, we can use the placebo to bridge between those other 2 treatments. So you can see in this schematic, you have solid lines indicating comparisons that were actually made in a real world trial. And then you have these dotted lines, which are essentially inferred based on this shared connection with a placebo reference arm. So where does model-based meta-analysis come in? Essentially, model-based meta-analysis extends network meta-analysis. It shares that approach of comparing treatments that weren't necessarily studied together in the same real-world trial, but have a common comparator. But what is added is pharmacological aspects to the model, such as dose or time dependencies, that allow you to use like more of the totality of the available data. So for example, a network meta-analysis, historically, you would filter the data such that you might only be comparing label doses of a drug at key time points. But with model-based meta-analysis, you describe the dose dependencies using something like an Emax model and time dependencies such as the onset of effect or potentially even loss of effect, so that you can incorporate time points that are maybe not pivotal, but very informative. So let's have a quick look here at what some key features to a model-based meta-analysis model might look like. So here you can see a time course of HbA1c change from baseline in a particular treatment model, arm of a study, and this is placebo adjusted. So the overall effect has essentially in this model 3 key components, 2 of which are time dependencies, 1 of which is a dose dependency. So the dose dependence should seem fairly straightforward in that you've got a simple Emax model with an ED50, which is the dose at which you get 50% of the maximum effect, which is denoted by Emax. That is in turn multiplied by a sigmoid function of time, of which ET50 represents the time at which you get the 50% of the maximum possible effect of the drug. But you can see in this particular time course is that after about 24 weeks or so, 20 weeks or so, that the HbA1c is starting to rebound, which might be due to loss of efficacy. So there is also a term for that, which is just a simple power function. Of course, the T-dur, the exponent is negative because we're going to have essentially a decrease in effect over time. So these, not every model has these features. These are just sample examples. So one of the things that we offer within my company are databases of clinical outcomes, and that, you know, one of the aspects of that is, you know, any analysis is only as good as the data upon which it is developed. And to have a robust database, you have to, you know, follow those steps of the systematic review that I talked about just a little bit ago. And also you have to have periodic updates. And so just as with, you know, the analysis having certain key steps, the development protocol for the databases also have to have that, particularly to ensure that you're not getting an unrepresentative sample of the information that's available in the public domain. So one way we help to control that is to have database protocols define how our databases are developed. So, you know, essentially and this again ties back to the systematic reviews. Before you develop a database, you've got to sort of you got to sort of define what is of interest and what is not of interest. So for instance, you might say, I'm only going to include randomized controlled trials in my database. And so a priori, before you even dig into the data, you should have some idea of what you intend to include and what you do not. And as I mentioned earlier, systematic reviews and meta-analyses usually starts with a research question. And so that, you know, starting with that could have, you know, potentially has a big impact on what your database protocol looks like. And then from there, there's maybe a more detailed specification of what variables you're going to capture in the database. For instance, are you going to collect body weight? Are you going to collect measures of renal function or disease severity? All of these, you know, you may want to have them be accessible when it comes time to do the analysis. So I'm going to pause there for just a moment. It looks like I have a couple of questions, or at least one. So I say here's one. Is there a repository of documents and learning resources where one can learn how to conduct a systematic review? Yes. The answer to that is, and I'm trying to remember the name of it. There is let me put a pin in that one. It'll come back to me, but there is, I got it. It's the Cochrane guide. These folks, they have put together. So Cochrane is the institute you want to look up. They're sort of the gold standard for how to conduct, robust systematic reviews. So that's Cochrane, CORCHORAN is the institute you want to look up to have some resources around that. All right, so I'm now going to talk about some sample applications of model-based meta-analysis. And the therapeutic focus here will be in rheumatologic diseases. There's some very good examples here, and some of the earliest, one of the earliest model-based meta-analyses was in fact in rheumatoid arthritis. So this paper was published in 2011, so a mere 11 years ago. It was the lead author was Jaap Mandema, who essentially is one of our experts here in CERTARA in the field, and along with several other co-authors. And this was essentially looking at AACR Response. So AACR is a sort of multi-panel diagnostic tool that was defined by the American College of Rheumatoid Arthritis. And basically, 20, 50, and 70 are targets of measuring relative improvement in the score with respect to its baseline value. So the higher the target, the higher the number, the more stringent the target. So AACR 70 is harder to hit because that means you've had a 70% improvement from your baseline status. And so you can see each of these panels represents a different therapeutic intervention. And then the x-axis is the dose regimen used. And so what the authors focused on here were the key time points. By that, I mean the pivotal time points in the trials, which were at greater than or equal to 12 weeks, which essentially you're at a dynamic steady state there with regard to your response to the drug. The analysis included 50 trials in total, representing over 21,000 patients and 5 different therapeutic classes. So you can see here, this is a lot of information. Now, you may be wondering why you have the different sizes of the symbols here. Well, remember back to when I was talking about meta-analysis, and I said that not all studies are created equal. Some have larger sample sizes. And that's really what these bubbles are about, is that the larger the bubble, the more informational weight that the trial was considered to contain. And so bigger bubbles have more influence on the fit of the model. So next, in terms of application, in 2012, another set of investigators published a meta-analysis in which they were comparing a new therapy, an IL-1 beta inhibitor, versus existing therapies. And so it was Canakinumab was the drug that was being developed by the sponsor. And so they assembled a database of 37 trials, roughly about 13,500 patients represented, and again, about 5 therapeutic classes. But one extension these folks did relative to the Mandema et al. Paper was, instead of looking at just key, steady state time points, they instead incorporated aspects describing the time dependence of the AACR 20 response. Now, the results you see here is the Canakinumab compared versus placebo, which is the little gray time curve toward the bottom of each plot. And then these two comparator compounds, etanercept and adalimumab. The client's proprietary data, you can see that the little blue circles are their placebo arm data, and then the yellow squares are the observed data from their active treatment arm. And then you can see that the shaded region is what's predicted to be the AACR 20 response relating from their therapy with some uncertainty. And in both instances, their drug is not really performing as well as either of the comparators. So based on this, this outcome, the client was able to conclude that their drug, at least in this indication, was not likely to be competitive. And so they actually discontinued the program. Another application of model-based meta-analysis is it can be used to do clinical trial simulation. So again, this is a published example where a sponsor was looking to design a dose ranging trial. And initially they had considered essentially a 25 milligram, 100 milligram, and 200 milligrams given at 0, 4, and then every 6, and then at 16 weeks. And then as a even higher dose regimen, they decided to include a 200 milligram Q4 week. So they don't, basically is keep giving it every 4 weeks. So in this plot, essentially a model-based meta-analysis was performed. And then we did simulations of the plan design. And the 3 colors indicate the teal means that, and we also simulated many copies of that design, so repetitions. So if you see teal is the fraction of the simulated trials for which that particular dose arm was at the top of the Emax curve. And the yellow is the fraction of simulated trials in which that treatment regimen fell sort of somewhere between placebo and the plateau, the top. And then red is indicating that the percent of simulated trials where that treatment did perform similarly to placebo. So this is a dose ranging trial. What we want to understand is really what is the potency of this drug. And if you look at the 2 uppermost arms, they're performing almost identically, or actually you could say 100, 200, and then the 200 Q4 weeks are all very, very similar and not really very informative with regard to the potency of the compound. So based on these simulations, it was decided to drop the 200 milligram Q4 week and instead include a lower 5 milligram arm in the hopes of getting a better characterization of the dose response curve. So just a couple more applications, and I won't take but just a few more minutes here. This is an example of how you can use metadata to characterize the relationship between long-term and short-term response. Now here it's the same endpoint in that it's ACR 50, but you can see that there's a very strong correlation between what happens at month 3 and what happens at month 6 with respect to this endpoint. And in fact, that again, these are several therapeutic classes here, and there's no real clustering, meaning that you don't see a lot of blue squares sort of below the line of unity, and you don't see a lot of clusters above and below those lines. It seems like this relationship is fairly predictable across therapeutic classes. So one final application would be that assume for a minute that you've got an existing therapy for psoriasis, and you want to make a leap from psoriasis to psoriatic arthritis or ankylosing spondylitis. And it may not be that that dose, psoriasis dose, translates directly to those other conditions. You might maybe need to titrate the dose up or down. And so one application of model-based meta-analysis is to leverage other therapies or data from other development programs that have already made that journey, and thereby potentially identify what doses in the additional applications might be appropriate. So I'll just go back to the key points here. So as I have said at the outset, we've looked a little bit at how model-based meta-analysis relates to earlier forms of meta-analysis. We've talked a little bit about why you would want to do meta-analysis, what in that publicly available information is potentially cheaper and quicker to make use of than proprietary trial data. And we walked through a few examples of how meta-analysis, and particularly model-based meta-analysis, has been employed to inform drug development decision-making. So thank you for your time. I'm going to stop sharing my screen because I think Ayapa, you want to share some slides, right? Ayapa, I think you're muted.
Thank you, Dr. Lovern. Can you hear me now?
Yes, I can.
Thank you very much for an excellent talk. I think I would like you to give this lecture in our course as well, which is a very application-oriented talk. Thank you very much again for accepting our invitation. I would like to share
you again.
couple of slides. I think we have a couple of questions which we will take after briefly touching on my a day in life. Me and Dr. Macu thought our students and aspiring students also feel a little bit about how a day looks like in a pharmacometrician's role. Dr. Lovern's talk nicely puts a great tool within pharmacometrics, model-based meta-analysis. I think we use several of such analytical tools in our day job. So we thought we will provide a little bit glimpse of that, at least giving my own examples. Can you all see the slides now?
Yes.
Yeah.
Okay, thank you. So basically, pharmacometrician's life and job revolves around drug development and clinical pharmacology. Pharmacometrics is the quantitative wing of clinical pharmacology, also called quantitative clinical pharmacology. So in order to do some of those analysis, which seamlessly sounded easy to explain for Dr. Lovern, but that comes with a lot of knowledge and skills that one needs to conduct. That include pharmacokinetics, pharmacodynamic knowledge, clinical trial design information and knowledge, disease progression knowledge, modeling simulation tools such as software programs, R, non-mem, Phoenix, which is CERTARAs, and Pumas and some other in the similar region of analysis, and regulatory submissions, because you're dealing with drug development. The analysis that you're going to do are directly impacting the drug development programs for the companies who are developing the drugs. And some specific questions I thought I will put, that's a broader knowledge base one needs to have once getting into pharmacometrician's role. But here are some specific questions my teams and I am working right now at Pumas that might give an example of projects. What is the exposure response relationship in a pediatric rare disease? So in rare disease situations, you don't have too much data available. Even if you can do a clinical trial, being a rare disease, you need to have some of the literature-based support and natural history-based support, which are essentially linked to some of the analytical tools Dr. Lovern also showed. And characterizing a population pharmacokinetics for an oncology drug being developed for a specific genetic mutation. What would be the first in human dose of a drug based on minimum anticipated biological effect level? Characterizing disease progression using natural history data. How to justify pediatric dose based on clinical trial data from adult population. Developing Bayesian design dosing tools for an antibiotic and integrate with clinical decision making dashboard systems. And overall, job is directly impacting patient lives by working with drug development teams to help develop novel therapies for unmet patient needs. So I would like to talk at least two specific questions that I showed to give what is involved to get or to answer those questions. So one example here is what would be the first in human dose for a drug based on information we can get to understand minimum anticipated biological effect level. So in drug development, preclinical development part of the story before it gets into human, you do have a lot of data coming out of translational models from in vitro cell lines, ex vivo models such as human blood or separated peripheral blood mononuclear cells, mice disease models. From all these, we put together models of concentration response, which looks like a S-shaped curve, which concentration increase, effect increases, and it saturates. We call them Emax models. Maximum response, EC50 is a parameter Dr. Lovern said, a concentration at which a 50% response happens. So we kind of characterize these curves and estimate those parameters using modeling tools and come up with parameters of EC50s. In parallel, you'll also have toxicological data from a couple of animal models such as mice, rat, and monkey, which we will use the data to build models to inform first in human dose. So from this response curves, we derive something called EC20, which is 20% effect level, which is minimally effective. We don't want to target 20%, but first in human dose, the goal is to have a safe dose to be given to evaluate the safety and tolerability in a first in human study. So we can derive this EC20 from these curves and pick the EC20 that gives the lowest concentration or an effect, which is called minimum anticipated biological effect level. Using this EC20 and combining with a modeling tool called allometry, which is based on body size and PK parameter relation, we can use the toxicological data to predict human clearance and volume or distribution of that drug. And using EC20 and PK parameter predicted from animal data, we can come up with first in human dose. In parallel, we'll also use FDA-derived no observed adverse event levels derived from toxicological data and derive human equivalent doses and maximum safe dose. Of all these algorithms, whichever gives us the lowest dose, we will pick for first in human. So this is the overall way of mechanics goes in, which building the models and deriving the parameters and putting together a justification for that question. All this needs some PK/PD analysis skills. Second example is how to justify pediatric dose based on clinical trial data from adult population, which is very common thing for any new drug development we need to think about. So this is a typical population PK curves in a group of people. If you give the drug and measure concentrations, they decline over a period of time after giving the dose, which we characterize by compartmental models. In PK course, most of you probably have seen them. And in case of clinical trials, most of them are done in adult population in the initial phases. We would like to come up with a dose for pediatric population. One way we do that is we build a model using compartmental methods, which boils down the data to relationship of clearance and volume or distribution, important parameters of drug disposition, to patient covariates such as body weight. And we use these relationships to simulate in a pediatric population using appropriate body weight distributions. This is a very simplified way of it. It could be more than just body weight. You may have age as a covariate, or you may have, depending on the pediatric population you're dealing with, if you're going all the way below neonates and all, you may have multiple other covariates of interest which will be used. But overall framework is you use adult data to develop models and use simulations appropriately to inform your pediatric dosing regimen. Again, this involves some obvious knowledge of PK/PD first, and then software skills such as R, non-mem, or Phoenix, etc. So hopefully these two studies give some glimpse of what happens in a pharmacometrician's job, and how does UNTHSC certificate would be helpful. We designed it in a way to build foundational knowledge in PK, pharmacokinetics, pharmacodynamics, and statistics, which are all essential knowledge and base you need to conduct such analysis. And also it builds foundational software skills that are required in addition to your knowledge, such as R, non-mem for data management, model building, and simulation capabilities. And overall, it gives understanding of clinical pharmacology and its role in drug development and applying these quantitative tools through case studies. So with that, I would like to open up questions for Dr. Lovern, and if you have any for me, then we will go to the closing remarks to Dr. McHugh. So Dr. McHugh, Dr. Lovern, could you turn on your videos so that we can start taking questions? Please type in your questions in the chat box. I think given so many people, it may be difficult if we start speaking. So I have few here. One from our student Ellis Vo. She liked your description, named after the father of evidence-based medicine. How poetic. But the question is, how do you account for potential differences in participant populations or methods among those papers in the meta-analysis?
So yeah, I actually put a response in the chat, but I'm very happy to speak to that as well. But essentially what you do is you treat trial level characteristics similar to covariates that you like you would handle as individual level characteristics in like population analysis, right? So if you think about like a pop PK analysis, we try to account for differences in exposure, usually with regard to individual characteristics such as, you know, body weight or gender or race. In meta-analysis, because again, we're working with summary level data, you know, we can look at those covariates, but some of the things that usually come up are more trial design features. So for instance, you might have more than one definition of a particular endpoint. And so you might include endpoint definition as part of, you know, a covariate that you would consider in the analysis. You might also, other key examples, baseline. Baseline is a big one, where if you're you have a study with the more severe status at baseline, you might actually see a bigger effect of the drug because there's more room for improvement, so to speak. So we, you know, it's analogous to the sorts of things that we do in, you know, individual based pop PK analysis. Having said that, you know, many times there's significant study heterogeneity that you can't really figure out where it's coming from, but you still want to characterize it, or in which case we do apply mixed effect modeling approaches in a meta-analysis context, just as we would if you were analyzing individual level data.
Okay, thank you. Thank you for that explanation. I think we are up to the hour now. I would like to thank, I think Dr. Lovern, thank you for answering the questions on the chat already. So the recording will be available on the university website. So please visit as you like to see the lecture again. And with that, thank you again, Dr. Lovern, and over to Dr. McHugh.
Well, thank you, Dr. Chattopadhyay, and I'd like to thank everyone for attending today. In closing, I'd also like to share with you, well, thank you, Dr. Lovern, first for an outstanding presentation. And I'd like to share with you our pharmacometrics certificate program. And so in this slide, you'll see that the program encompasses 3 semesters, starting in the fall with 6 semester credit hours, spring 6 semester credit hours, and then 3 semester credit hours in the summer. I have also put in our URL for our website on the program in the chat. And so you can find out more about the program by visiting that URL. And thank you again for joining us today, and have a great day or evening wherever you are. Thank you.
Thank you, Dr. McHugh. I think I know I just have seen one question someone has, Luong Wong, they want to ask on the program. Can you please ask your question, Luong, or type in your question here? Okay, is the pharmacometric certificate critical for someone who has a master's in pharmacokinetics? Also, is there an opportunity for PhD students at the university? I can take this. At this stage, we don't have PhD students for this particular topic. The first question, is pharmacometric certificate critical for someone who has master's in pharmacokinetics? I think our certificate has been helpful for students who have some skills of pharmacokinetics, but don't have a systematic learning of all the skills, that knowledge that requires in PK/PD and statistics and software. So in a way, it's a overall global understanding for the foundations. So depending on the person's skills in modeling simulation, the utility of the certificate goes. But I would think that a typical pharmacokinetics master's, it is not focused on modeling simulation. Probably the certificate course will give that feather in the hat to to get the skills needed. I'm happy to look what you have done. If you can send your information so we can give a better advice on what you need.
And I see one other question here. And so please visit our website. There is a link to the cost of the program there. And I put that website on the chat.
And one question is, after graduating from the course, can one become a pharmacometrician? So pharmacometrician is a very broad term. Mark Lovern is a pharmacometrician, and a fresh graduate student coming out of school will join into pharmacometrician role. So the band matters, the knowledge skills differ quite varied. It's like a statistician with 20 years experience versus fresh graduate. So it depends on the level of education the person is having. In our experience, what we have seen, if a person in clinical pharmacology already in industry, they're taking certificate course, they're easily transitioning into pharmacometrics roles. If a PharmD student or a fresh PhD student is doing it, they are getting into good fellowship programs. So it depends on where you are in your education, and the certificate can put that feather in the hat again to move into the trajectory of pharmacometrics roles. I have a master's in pharmacometrics, but would like to learn MBMA. That is Dr. Lovern's, I think.
Oh, sorry.
He's saying, is there a workshop to learn it? Does CERTARA offer any workshops right now?
You know what? I don't think we have done workshops for some of our clients, you know, at their request. I don't know that we've done a public, you know, open call workshop, but that's a good idea. So perhaps I could take that up. We have a guy that we hired last year, Matthew Zierhut, to facilitate the development of a practice area in model-based meta-analysis. So maybe I will suggest that as a you know, an additional offering we could provide.
Thank you. Maybe last one, Dr. McHugh, I know we are off over the time now. I'm studying epidemiology. Is there a chance for me to work as a pharmacometrician? Yes, pharmacometrics is a very broad field that people with many different skills come in, and they use their core areas. Dr. Lovern is a biomathematician, as you've seen, and there are a lot of pharmacists, pharmacokineticists, pharmacologists, mathematicians, statisticians, and I would think one needs a little bit of conditioning what they don't have in terms of pharmacology and PK/PD, especially epidemiology or a pharmaco non-pharmacy type background. But I think our certificate gives that cushioning of overall knowledge base. Happy to talk to you more, Luang. I think please send us an email, or we can set up a call with you to go over more of your questions. With that, Dr. McHugh, I think I'd like to thank everyone again for joining, and we can close the call now.
Thank you very much, everyone. It's been a pleasure to have you.